In this case, we will discuss about Fragile X syndrome. So, this Fragile X syndrome is related to weak and elongated, and immature synaptic connection in brains, and it has been presented by this figure. Here, the normal connection between two nerves has been shown, the synaptic connection. However, in case of Fragile X syndrome, this this synaptic connection get elongated, and that might lead to several problems in the brain, such as the motor connection will get affected, and there are other problems also. So let us understand what is the mechanism and why this Fragile X syndrome occurs, why or how this Fragile X syndrome occurs. So in case of normal condition, the repeats of CGG is less than 45, and we have understood that what is C, what is G, what is A, what is T in one of our previous videos. These are the nucleotides that are present in DNA, and DNA is made up of these four nucleotides only. And when the repeats of this CGG are less than 45, then it is considered as a normal case. And this repeats comprises of finally comprises of gene, that is FMR1. And when this first is transcription of this gene leads to the formation of mRNA of FMR1, and after transcription, the translation occurs. As we know that this is the central dogma of the cell. So after translation, the FMR1 mRNA converted into FMRP proteins, and the function of FMRP proteins is to control the overexpression of mGlu-R5, as it is shown by here, this arrow. And because of this, mGlu-R5 will remain within limit, and it will prevent the elongation of the neural network or disruption in the synaptic connection. And let us understand, there are two kind of conditions that can occur in Fragile X syndrome. First one is the full mutation. So what happens in the full mutation? The repeats of CGG are greater than 200. So because of this, because of higher number of repeats, there is a more possibility of methylation of these repeats. And as the repeats are now methylated, now they are not in a condition to be transcribed, as they will not be able to transcribe after methylation. So as there is no transcription occurs, there is no formation of FMRP protein. Because of this, there is no control of over mGlu-R5 proteins. So this leads to overexpression of mGlu-R5 protein, and this might cause uh, this will cause the elongation of the uh, this spine, and that might lead to problems in the brain. And this things has been studied in the mice with a knockout of FMR1 gene, and this kind of problems has been observed in the mice. And there is a third another condition which is known as premutation. So what is the condition of premutation is that the sequence of CGG is between 55 to 200, and in this case the repeats of CGG are higher than the normal but is lower than the full mutation. Because of this, the repeats are not in a condition to get methylated, and thus during the translation of FMR1, it will lead to excess amount of FMR1 mRNA production because the repeats are high. And as we know, and the study, and it has been predicted that FMR1 will interact not only with FMRP proteins but with other proteins also, and this excess formation of FMR1 mRNA will lead to excess will interact the other stock of proteins, and this might lead to depletion of proteins in the system for the normal function. And because of this, the several other problems might occur. And When this mRNA binds to the excess depleted excess proteins that are, or when the excess amount of FMR1 mRNA binds to the other proteins to which it has a interaction, this excess mRNA, the amount of mRNA that are left left for translation into FMRP proteins might also get affected. If the interaction is low between the proteins and the excess mRNA of FMR1, then high amount of FMRP proteins are formed. Otherwise, if the interaction is high, then the less amount of mRNA are left to convert it into FMRP. This will lead to low FMRP productions, and low FMRP protein productions will lead to less control over mGlu-R5. So this will leads to further elongation of weak spine. It might also lead to other problems such as LTD, that is long-term depression, that has been observed in hippocampus and cerebrum of the mice that has been knocked out with that has FMR1 knocked out uh, mice in which the gene FMR1 has been knocked out. So that's all for this video. Thank you for your time.